So welcome to this afternoon's seminar, and I'd like to start with a land acknowledgement. So we respectfully acknowledge the island of Newfoundland as the ancestral homeland of the Mi'kmaq and the Yaqut people. We recognize the Inuit, Nunatsiavut, and Nunatukavut, and the Innu of Sheshashi and Nakoshish and their ancestors as the original people of Labrador. We strive for respectful partnerships with all the people of this province and move forward and honor this beautiful land together. I'd also like to acknowledge the people of Nunavut and Nunatsiavut, in whose territories we were working on our cruise this summer. We were in the offshore marine territory, and but I'm particularly excited about developing and ongoing collaborations with both of those territorial or indigenous governments with regards to research and conservation of the waters. So before we get started, I have to tell a story. I apologize. We were on the ship, so Max and Barbara and I were on the ship with, as well as several of the students who are here in the room today. And we got back in the middle of August, and I was preparing supper in the kitchen the next day, and I heard the broadcast come off. And I thought, oh, I know I'm home. And then I thought, boy, I bet Janie would be interested in what we are doing. So we set up an interview with the fisheries broadcast, which Ratana mentioned in one of her classes. And because of that interview, I was asked to, well, would you like to present to the geography department about what we did this summer? So this is kind of a what I did with my summer talk. But it's more than that, as you will see. So it's a three-ring circus. So Barbara is going to present first, and then Max, and then I. And you will see a little bit of overlap between what we're doing and where we are pointing to what each of us are presenting about. But one of the most exciting aspects of working with ArcticNet, and Trevor can attest to this with his long experience with ArcticNet, is that it really is a collaborative network where it's not just people in their own silos doing their own projects, but really crossing disciplines, whether that's on board the ship or across the network, working even across science and social science disciplines. So now it's time for me to be quiet, and Barbara should start.
Thank you. Hi everyone, thank you so much for coming. Can you hear me well? I'll try to do this in my 15 minutes being allocated for me. So as Evan mentioned, I'm Barbara. I'm a research scientist at the Department of Fisheries and Oceans in St. John's, and I'm also a MUN alumni. So I did my PhD here 2 years ago, and I'm one of the lucky ones to continue working on what I love, and I really work a lot on cold water coral and sponge ecosystems in Newfoundland, Labrador, and also the Baffin Bay region. So this is the computer, right? Yeah.
No.
No?
Oh,
it's this one, right? Okay. So today, as Evan said, I'm gonna get started, and then Maxime will talk, and then Evan will talk after. So we did a little bit of a part 1, 2, and 3, and we'll see how we can connect those different parts. Just put a little snapshot of our cruise report. The lists our principal investigators, and Maxime was our chief scientist, just as an acknowledgement of all the collaborators that participated in the preparation of the expedition this summer. So I'm going to talk a little bit about the exploring part. So the talk is about cold water coral and cold-seep ecosystems, exploring, finding them, and let's see what you find. So I don't know if all of you know, we have at least 70 different species of cold water coral in the Newfoundland region alone, and they are vulnerable. As you can see in these photos, very often we see corals attached to hard bottoms, to rocks or little pebbles or big boulders. Sometimes we see them on soft sediment, but they're vulnerable, they're fragile. If we knock one over, they are not going to come back and begin growing again, they're going to die. So they're fragile, they are very often long-lived, hundreds or thousands of years. They are slow growing. And when we're studying them, we've been more and more, with the international community looking for alternative methods to go and survey them. Talking about cold water corals, but very often deep water corals. So we've been working a lot on getting access to deep water environments using cameras. So it's our seafloor imagery, it's one of our main tools of choice. And so today, we're going to talk a lot about that, about how we did the survey last summer that Evan was talking about, but I'm also going to go back to some of the past years, some of the surveys we've done, again, exploring and looking for these coral habitats. So the ship you see there, this is the Amundsen. It's a Canadian icebreaker. That is the platform we use, that we've been using for many years to conduct our research in the Arctic. And I just put those little circles there with different types of methods and techniques we've been using. So as I mentioned, seafloor imagery is one of the main ones we're trying to use more and more to survey cold water coral habitats. But we use a lot of these different methods, all of them integrated, and that's very important for what we do. So seafloor mapping, oceanographic data, data that we might have on fishing, scientific fishing, bycatch, local knowledge, very important too, and even sediment coring. So I think we're going to try to talk a little bit about these different things as we talk, as we give different examples of what we saw. A lot of focus on seafloor imagery on my side. So as I mentioned, I was going to go back a little bit. So I started doing these surveys in 2013, and we used this remotely operated vehicle, an ROV, for the first time in 2013, at least my first time using it. And we've been using it until 2018. So it had high definition cameras and could do good surveys of the seafloor. But it gave us a lot of headaches too. It was sometimes a little bit problematic, but we also got very good data with it. We got to see sites that we had never seen before and nobody else had. So this is one of the tools that we've been using. A second tool that we've been using to collect seafloor imagery, photos, videos, well, basically videos, is a drop camera system that you can see here. So this is a camera mounted on a box core, a sediment core frame. And this system can go, we've been collecting data up to over 2,000 meters water depth. And so that ROV is the one that we stopped using in 2018 because it was too old. Then Amundsen Science, who manages the science in the Amundsen, now have that one that you see. And you might actually, okay, the mouse works right now. You can see the little coral here that it had collected with the robotic arms. So these are the main tools that we've been using in the past years to collect this imagery of the seafloor in this region. So this map is showing basically the little squares you see are the positions of sites we visited last summer in 2021. And you can see that we went from Makovec Bank in Labrador all the way up to, this mouse is a funny thing, all the way up to Scotland here. And I'm going to talk about some of these sites. I know that Evan is going to get into detail on some of them, and he might probably talk about some that I'm not going to touch because we don't have the time. So I'll begin with Disco Fen. This site is very special. We've been visiting Disco Fen for a number of years now, and you'll see why. Well, actually, I'm going to begin talking and then I'll show the map. So in 1999, there was a scientific trawl that, bottom trawl that took place in Disco Fen and recovered a large amount of a type of coral that we call bamboo coral. And the amount was so big that we knew that there was something special at this site. So in 2013, we had an opportunity with that ROV, the first one I showed, to go to that site and do a video survey for the first time and see what the bottom looked like. So we knew probably lots of coral, but we had no idea of what, right? So we went in 2013 and then we planned, this is the map of our cruise plan at the time. And what you see here, the main thing to pay attention is, you see this red line here. This is basically the start and end position of that survey. So we planned this yellow zigzag line here, so we can see actually, that we would cross that trawl path and see if maybe we would find those corals that we had never seen in this area. Something went wrong with this photo, but basically it's just to show you 2013, not a long time ago, but you can see these screens are not very recent. But this is the system we had at the time, and we actually got to visualize the seafloor, and we were very happy about what we saw. So that was the old ROV. Just want to show you what it looked like. So in 2013, this is the video of the very dense, this is a very dense coral hotspot. These corals are forming these very large patches in a muddy bottom as you can see. This is about 930 meters water depth. And it's very, very dense. We don't see that often. And this is a species that usually we see attached to rocks, not often found on a muddy bottom. So they do have an adaptation to be able to anchor on soft sediments. So we were very impressed about this site. You can see the quality of the camera is good, but it's not great. It wasn't high definition in 2013, and then things got better over time. But this is something that we were very, very impressed about. And this site now, it is part of one of our Canadian marine refuges, so there is no more commercial fishing inside this area. So Disco Fan is one of our favorite sites because we discovered this, and then we went the first time and we don't know what you're going to find, right? So the scientific questions begin to come after we see it for the first time. So then we go back with more defined questions. And I think Evan is going to give some good examples of how we've been doing science in this region. Just want to show you, so this is a video from this year, and it's still very similar. It's very bright, I don't know why the video is kind of showing like this, but you get it. It's very dense, and you can see there is a lot of material in the water column too. Just very similar to what we saw last year. But this is another location, a little bit different from the sites that we knew. And this is one of the questions we have. We're looking at this and we keep surveying, and we go and we see a patch of mud and then we see them again. So we don't fully understand how they are distributed. And one of our questions is, well, what is the spatial distribution of these patches? How far do they go? What are the depth limits and that sort of question. And because when you see very dense corals like this, we clearly, think of the importance of the habitat for the species. If you're a little animal looking for a place to hide, well, you can hide them, let's see. Basically it's what I just said. It's our question of how far do these forests extend, and we still don't have a good answer to that, but we've been going back to this site a number of years now. Okay, the second example I wanted to give you is a survey that we did in 2018. Maybe you can see it's actually here in Greenland. Has a little L for Lophilia. So Lophilia is a type of coral. And it's a very interesting story too. You see, the other example I gave, the Disco Fan example, was based on some data that somebody came and said, hey, there's something here, right? It's not when we select a place to go to our survey, it's not, we don't go there blind. We need to have some indication of something to go. And the Disco Fan was that data on the scientific trawl. In here, we had a different type of information that was given to us, which basically was, I don't know if you guys are familiar with this. This is a CTD rosette stamper. It goes down the water to collect water. So it has a bunch of bottles. And actually what happened here was what we call a rosette or CTD bycatch. It wasn't a net, it was the CTD rosette. And we don't have a photo of what happened, but basically this coral here was found in the rosette when it came back during a survey just looking for water. This wasn't our team, was other colleagues at the Nova Scotia a few years back. And then we call it was the CTD bycatch. It doesn't happen that often. But then it was a very important finding because there was no knowledge of the presence of that coral at that location, right? And Lophilia is a very, very well known now cold water coral that forms reefs. It's a cosmopolitan species, so it's a big deal finding Lophilia, and it wasn't the only species found. But anyway, so we decided to go there too. And this is another map showing how we did our planning. Just to give you an idea of how we tried to put things together to select where exactly to put our cameras. It was also an ROV survey, and this is one example of the plan we had. As you can see here, there's a little star. The little star is the position of that CTD bycatch. And then we did a little bit of a planning to cross it, but then to survey different types of environments, beginning at 1,100 meters, ending at about 700, close to 700. The final plan looked a little bit different because, the conditions sometimes are more challenging, but we still got to see what those cliffs look like. It was like a vertical wall, very steep and challenging to work with. But this is a photo of what it looked like. And if you're not familiar with this, well, all of these are colonies of this Lophilia coral, And then we had all of these clams. We had these other species of coral, which is a gorgonian, a large one. The one that was in the ROV arm, and I'm going to show this one again, called Primnoa. And you can see what the environment looks like. It was quite impressive. And we could also understand how the City de Rosette got caught. I mean, got a piece of the coral because it was coming back up, and then they were lucky that they didn't lose the rosette. Need to go faster. More corals. You have a bunch of soft corals here, and again, this gorgonian coral. And there you go. So then I'm going to go tell this story before my time is up. The Makovic story. Maybe some of you saw this on CBC. We did a little story about how we found a biodiversity hotspot here in Makovic. So the story in Makovic is that you see this little polygon. So there was a fish harvester called Winslow Bartlett fishing in the area, and he knew about the presence of corals in this region. He was collecting the bycatch, and then one day, and Evan met him a few years ago, and then my colleague Vonda Hayes at DFO actually went to see him, and he showed her collection, his collection of corals. So really showing, yeah, that's proof of what I've been seeing. And these are all corals that were interested because they are vulnerable, they are long-lived and that sort of thing. So then Mr. Bartlett, he basically on a map showed more or less the position of where he was finding these corals, and it's that little polygon that you see that our colleagues at DFO produced. So then last year, I think last year, 2019, the J you see there, that's for Joey. So Joey Egnatok is another fish harvester from Maine. And then Joey told my colleague David Cote at DFO, look, there seems to be another place that could be of interest with the presence of corals. He had some indication that the area had corals at a cliff. So we did a bunch of surveys in 2020. You see all the little dots looking for potential indication of a hotspot of diversity in all those corals. We didn't find anything. But then that year we collected bottom mapping data, and then we used that in our planning. So what you see there is a map of our multi-beam data. You see 4 multi-beam. And what you see in black basically are areas of very high slope, over 50 degrees, that's what I had. So we did our planning based on so many different aspects, but really the indication from the fish harvesters and us studying the multi-beam and our knowledge of the distribution of corals in the region really helped us to pinpoint where we wanted to dive. And what you see here are the transects that we did with the ROV, the new one this time. And this is a video of what that cliff looks like, and was also a very nice, I don't know if I should say surprise. We were just very happy to see this. This is a coral that is also, can be hundreds of years in age. Primnoa, some people call it the popcorn coral. It's a gorgonian type of coral. They can grow very tall, and they are habitat for different species. You can also see a number of sponges in this area. So this was very, very impressive to us. And this is not very far from land. It's about 20 miles from Makovic. And this is about 650 meters. Yeah. And this is just another photo, not a photo, sorry, a video, just showing you how we can sample a little piece of this specimen, or the whole specimen in some cases. You can see there was a little fish in here. Yeah. So this was Makovic. Should I keep going?
Yeah.
3 minutes. Okay. I just wanted to now mention, because we're talking about cold water coral habitats, but also cold-seeps, hydrocarbon seeps. So I just wanted to mention here in satellite thing, we had a dive looking for coral habitats and other things like this, and we ended up finding the presence of bacterial mats, which are these white things you can see here in the seafloor. And so this is another indication of the presence of a hydrocarbon seep that had not been identified before. So we were very happy to see this. And now it's another site that we know that we should go back and do some more research. We did some sampling here, but we didn't know about it, so we were very excited to find this. And then to finalize my part, then going to Scotting Lake is another example of a cold-seep location. So basically what you see here again, the white is microbial mats that we have on the seafloor. And at this side, you can also see bubbles of methane coming from the seed form. And basically, the identification of this site was also based on different, talking to different people. And one example is the presence of oil on the surface indicated that there was something going on at this site. But then some of our colleagues started doing, deploying the rosette that I showed before to collect water and looking at the methane amount in the water. And then we would look at some of these results. Seen here, for example, these are the sites that we were interested in because it has very high concentrations of methane. And then we did some surveys with the cameras looking for these areas with high methane, and sometimes, well, not sometimes, we ended up not really finding more of this, but it's also important to know when you don't find something, right? So just a quick video of, again, this is what is in white is the microbial mass, and the ROV is basically sampling with the push core. And we have some colleagues with, that are working on microbiology associated with these cold-seeps in these mats. Well, Evan's going to talk about this, so these are just some photos of the region, but I'm going to skip because I don't have a lot of time. Just wanted to mention that we found these crinoids. It's a huge crinoid field in that photo you see there on the corner. And it's also another benthic hotspot. Nice.
Thank you. So, as Evan mentioned, there's one important part of that of this kind of research is that it's really multidisciplinary. So, Barbara had a really good introduction on the benthic habitat and the cover forest. And then part of the cruise was also to, and of the study was also to look at the connection between pelagic and benthic. And here I'm going to present some of the work we've done on pelagic ecosystems, so what's happening in the water column. What is interesting is that the area that we covered really covered what we call the Atlantic-Arctic gateway. So we really went from the North Atlantic up to Baffin Bay, which is really the Arctic realm. And then by doing so, we could really compare these two and contrast these two ecosystems. And just as a general introduction to that, if we look at the schematic of both ecosystems, the North Atlantic versus the Arctic pelagic ecosystem, one thing that is striking is that it's the further north you go, the less redundancy you have in each of your trophic levels. So you have less species as you go further north. If you just compare the number of documented species in the North Atlantic in terms of marine fish, you have 318 fish species that are documented for the North Atlantic versus 229 for the Arctic. Of those, you have 137 that are endemic versus 48 that are endemic in the Arctic. So you really see that one characteristic of higher latitude is that you have less species but are already specialized for their environment. If you compare that just as a reference, in the North Pacific you have 482, so you see about half of them in the Arctic. In terms of marine mammal, it's also the same, so you have 48 extant marine mammal species in the North Atlantic versus 35 in the Arctic. So again, less species, less diversity. One thing I'm going to focus a lot here, so the part that we're looking at with the pelagic is really this part where you have, and we're looking a lot on these small pelagic, so mesozooplankton, macrozooplankton, and forage fish. And these species are important because they funnel the energy between lower trophic levels, so from the primary production, up to larger predators such as piscivorous fish, marine mammals, and seabirds. So that's really what we were looking at during that study. Then again, you have more redundancy in the North Atlantic because you have species like Capeland, herring, you have mackerel, sand lambs, versus in the Arctic you only have one or two species, the main one being the Arctic cod. That's usually a key species funneling the energy. So if we look at the secondary production, which is the smallest animal we're looking at within our group for that study, the main species that you're going to encounter in the high latitudes are the copepods, very abundant ones. These are large copepods. So you have the Calanus hyperboreus, Calanus glacialis. In the North Atlantic, you have Calanus remarchitus as well. And these are the cows of the sea. They are really the grazers, very abundant. And they store the energy from the primary production in the forms of lipids. So you see these lipid sacs here. You see really fat, really big steaks that are available for the predators. Then you have some of the other species that we catch in our net. And so those are a few millimeters to 1 centimeter long. These one will be in the order of centimeters in terms of size. So you have the abundant Themistolibellula, the very voracious. So these animals are very, they're really survival. We catch them in the nets and they continue feeding and then do cotton feeding. You have the krill, which is very abundant. You might have heard about them because they're the most abundant animals in the world. But one thing to remember is that the abundance is mainly in the Southern Ocean. So there are very the main species, versus in the Arctic, their importance, relative abundance is lower. The main abundant species in terms of zooplankton are the copepods. And then we have a lot of gelatinous zooplankton as well. So large Scyphomedusa and then smaller gelatinous, for instance, metaphors. Well, these are the kind of animals we catch. And one thing that is important to know for that ecosystem, both for the North Atlantic, Labrador Sea, and the Arctic, is that there's a trend towards smaller species, most likely because of climate change. And then when we hear about the lower productivity on the shelf, in Newfoundland and Labrador shelf, and in the slope, one of the part of that lower productivity is because of this switch in the size of the zooplankton that is available for the predators. So we're going from these big steaks towards small popcorn. It might be more popcorn, but overall you have less energy in the system. Then if we look at one trophic level above, we're looking at the pelagic fish, forage fish. The diversity, as I said, there's a few hundred species that are in these regions. However, if you look at pelagic, it's only a few of them. Most of the diversity is really at the bento-demersal part of the ecosystem, so close to the bottom. But if you look at the species that are in the water column, especially at those small forage fish that really funnel the energy as I just described, you have about 3 species in the North Atlantic that are of high abundance. The capelin that most of you know if you've been long enough in Newfoundland. And you know when the capelin is rolling, when it arrives, it really triggers life in the ocean. Yes, when the whales are arriving, seabirds colonies start being there, and then you have also the Atlantic cod that is coming. That's when the recreational coastal fishery starts, and then tourism also. So you see that it brings a lot of life with these species. So very not trivial species to understand in terms of ecology. You have a bit of redundancy. So there's herring a bit further south. If you go in the southern part of Newfoundland, more towards the New Brunswick and then the northeast US, that's the main forage fish. And you have sandlands that are sometimes often overlooked, not because they're not important, but because they spend part of their time hiding in the sediments. So they're not as easy to catch and see. That's for the North Atlantic. If we go towards the Arctic, but also Labrador Sea, as I mentioned, we have more of these Arctic cod, Borrowedicida, and a very closely related species, the ice cod. Even though they're called cod, they are gadids. They are more like capelin, so they're smaller, so they're really forage fish, not like the large piscivorous Atlantic cod. So what we did during that cruise, so we deployed a whole suite of different nets. So we have stratified zooplankton nets to look at the vertical distribution of the copepods and different zooplankton. We have what we call the double square net that is towed at the surface to catch the larvae of the surface, fish larvae. So and then we have a bottom trawl to catch the fish that are close to the bottom, and a mid-water trawl to catch these fish living in the water column. So these are the kind of nets that are deployed from the side of the ship, as similar to what Barbara mentioned with the drop camera. So these are kind of fish that we caught during the last cruise. So a lot of them, as you see, are the juveniles or larvae. So these are wolffish, the big wolffish that you might have seen with the big teeth. Well, that's what the babies look like. Not as impressive. Baby skeletons here, so larvae. We have squids there, Gonatus fabrici. We have snailfish here, and one species that I'm going to come back to, the myctophids, the glacial lanternfish, very abundant in Labrador Sea, even though you might not have heard about that, probably the most abundant fish there, myctophid. And then the Arctic cod, which I keep coming back. Another tool that we were using to study these ecosystems are fisheries acoustics. So on board the Amundsen, we have broadband echo sounders, which is similar to what you're going to go for cod fishing, for instance. If you go with a small boat and you look at what's under your boat, it uses sound to see where the fish are. So it's basically the same system that we're using at different frequencies. And so that really looks at the sound beam under the vessel, that looks at different distribution of fish. And how we visualize that is with an echogram. So you have on the y-axis the depth, so here from 0 to 1,300 meters. On the x-axis is time, and if you're traveling that distance, so it's a transect of about 30 kilometers. And the more warm the colors are, the more dense the animals are in the water column. So you have the sea fur going from the shelf to the deeper basin. And that's a typical ecogram for the Labrador Sea. So you have this extended mesopelagic layer of fish and zooplankton all over the place. And when I call it mesopelagic, that's between 200 and 1,000 meters of water. And then we deploy nets in these layers, and interestingly, because we are in the Atlantic Arctic Gateway, we look south of what we call the polar front and north of it, which is about, the polar front is about the distance of Davis Strait. And then if you go south and you deploy your net in that layer that we see on the ecogram, it's almost exclusively these glacial lanternfish, the myctophids. As soon as you reach the other side, so when you are at the front, you have a bit of a mix of both, and then when you arrive in Baffin Bay, then you have Arctic cod. So you really have, you go from the more boreal kingdom to the Arctic kingdom just by crossing that front. It changes drastically. And that's one of the things that we were studying during that cruise, trying to understand what were the drivers of that change, and can it change with climate change, can it be modified? And why is it of interest to us? Well, the lanternfish, the myctophid like this, the glacial lanternfish, they are the most abundant fish stock in the world. So just the abundance varies, but it could be the estimates between 500 million tons to a few gigatons. To put that into perspective, the whole global fisheries landing per year vary between 90 and 97 million tons per year. So really not trivial in terms of abundance and extent distribution in the world's ocean. And of course, we have a growing interest from the fishing industry, at least in Europe, not so far in Canada. We can expect it's going to come because we have the largest fish stock in the world that is not exploited so far, except for marginal fisheries in the Oman Sea and Southern Ocean. So we're really trying to understand the ecology of these species. And then the same thing, so we are at the, so you see that's where the polar front is, so we go across Davis Strait. And what we see on the ecogram is that layer stops, it becomes really more fade. There are still fish, but really more fade, so not the different fish and not the same abundance. And that really coincides with the drop in temperature. That's one thing that we observed during that cruise. And trying to understand why is it so. So one reason why we think that the fish from the boreal areas are not adapted to the Arctic is because they conduct DVM, diel vertical migration, where they would go at the surface to feed during nighttime and descend at depth to hide from the predators during daytime. However, so that's what they need to do to feed, to survive. However, the further north you go, you have really cold water. So that's a profile, temperature profile from northern Baffin Bay. And you see that in September, you have temperature that are below zero. They are down to -1 in the top part of the water column. So that means that if you have fish that are not adapted to this cold water, like the lanternfish, it will be really hard for them to go to the surface during nighttime to feed because it's too cold for them. They don't have the anti-freezing proteins that the Arctic cod will have, for instance, that allow them to survive because they're really specialized for the Arctic. So that's one reason that we think why they're not going there, or not extending their range northwards. And the other one is because of the extreme light regime prevailing at these high latitudes. So if you see when we cross also around Davis Strait, Baffin Bay, we start having, cross the Arctic Circle, and we start having, at least for some time of the year, continuous daylight and continuous nighttime. We have the polar night and the midnight sun. And because of this continuous, at least for part of the year, continuous daylight, that means that, again, they cannot go at the surface during nighttime to feed, because then they're always staying at depth. They keep at the same isotherm, the same level of light for most of their life, really. So they don't go there if there's too much light. So because of these two reasons, that's what precludes some of the boreal species to go further north. That's really what the interaction between these two species, these two factors, and how it changes from one region to the Arctic to the other is really something we're trying to understand. And just so you know, I'm trying to recruit a PhD to work on that polar front impact. So a little bit of advertising. The last thing I want to present here is that one thing we saw also from the survey last summer, and that was one of the reasons why it's so interesting to work with people like Evan and Barbara, is that they really brought the geology and the geography and the bottom habitat, benthic habitat, into the research we're doing, which normally we would look more at the water masses because it's pelagic. But we realized, no, there's actually a link between both. They were interested in going in the area that Barbara described, the Scott Inlet. One reason is because it's a glacial trough. So it's deeper, on the region that is over the shelf. So normally over the shelf is not as, it can be productive, but you don't have this, especially further north, you don't have these warmer and denser, so deeper Atlantic water coming in, and they are warmer, more nutrient rich. But these are kind of hot spots on the shelf. That's what we realized, of bringing these nutrient-rich Atlantic water. And then by being there for other survey from other groups, we started looking at the acoustics, and then we saw these really dense schools. So again, this is the bottom here, surface, and the warmer the colors, the denser. But we arrived in the glacial trough, and we start seeing these really, really dense schools of, we didn't know why, but something was there. So very interesting. And then, again, working with multidisciplinary teams, they were deploying the drop cam. In that case, not the drop cam, it was a baited cam. So you have some bait here, and I have a camera. They deployed that there to see what's near the bottom. And as they were deploying it, and that's actually a bait that Barbara put for to catch some shrimps. I didn't know, but shrimp like cat food. And when it arrived at the bottom, that's the same place. You can even hear the ping of the echo sounders. So you know that's really what we're sampling. These are schools of Arctic cod, so very dense aggregation. So now we were able to relate the echograms and ground truth with what was seen on the camera, and we knew that the reason why they were there, it's most likely because of the really dense, of the entrance of Atlantic water in these glacial trough. And again, Evan and I will be recruiting a postdoc to work on this project. So just so you know. And that's it for me.
So now for part 3 of the free-range circus. So this is a picture of the same coral habitat that Barbara showed from northeast Southwick Bank. Notice these corals, the same ones we saw, Primnoa, the popcorn coral, but instead of growing on the vertical wall and hanging down, they are standing straight up on top of the boulders. So we'll come back to that in just a moment. Okay, so let's back up to the big picture. I have to speak with only one arm now. When we think about the Arctic and climate change, we typically think about loss of sea ice and the direct biological impacts of that, or the impacts on Inuit. But we should not forget about the impact of that on industry. In particular, the fishing industry has access to places that they did not have access to before. And the oil industry has access to places they could not go before as well. Similarly, oh, this is going to be annoying. Similarly, we have, since the moratorium in the early 1990s, the footprint of the Canadian fisheries has expanded a lot northward and into deeper water. And so we have been finding habitats by accident that we do not want to damage. And so that's where Canada's legal obligation to identify and protect vulnerable marine ecosystems comes from. And that's DFO's responsibility. Being from universities, we have to point the finger instead of being responsible for it. So DFO established the marine refuge. It's got a crazy shape because it's trying to find that balance between protecting the most habitat and also having the minimum impact on the fishery. So these gray dots there, that's all the fishing footprint. So they're trying to cut the shape of the enclosure around the fishing footprint. This little cutout here was a result of a negotiation between the fishing industry and an NGO. So the NGO said, okay, you can have that if we get 40,000 square kilometers of sponge habitat for the north. And the industry said, okay. So again, these other effective area-based conservation measures are very much negotiated entities. So one of the questions is, how deep do the corals go? So we started looking into deeper water using the drop camera that Barbara mentioned, but also this year looking at 2 dives that were in the 600 to 800 meter range. So, and in particular, I want to draw your attention to this one. So we're looking at multi-beam data collected during last year's cruise. We noticed this funny feature. We didn't know what it was and thought, that's funny, it looks kind of like a slump scar. But what would be causing a slump in that area? And so this comes back, so here it is with the shaded bathymetry, so you can see it more clearly. The green is where the ROV went, and the yellow is where it was actually on the bottom because of the strong currents, okay? So, as Barbara mentioned, it has what we think might be behind that slump. It's the fact that there's this cold-seep here. Here are the microbial maps that show the evidence of the cold-seep. The other interesting thing here is these crusts. These are called carbonate crusts or authigenic carbonates. When you have hydrocarbons bubbling out of the bottom, they interact with the seawater and it precipitates calcium carbonate, which makes it a discrete habitat for the corals. Most of the corals in this area were not growing on the crusts, most of them were growing on boulders. But it is feeding into it. The last thing to note is we did see some evidence of fishing inside the marine refuge. We don't know if the fishing line is more than 4 years old or if it is somebody who is either unaware of or disregarding the rules. So who cares? Why is this even important? Let's go back 10 years ago to a paper describing the distribution of cold-seeps in the Northwest Atlantic. So these dots here are all cold-seeps identified from satellite imagery. So radar sat, it measures the size of the waves. Where you have a hydrocarbon seep, the hydrocarbons actually dampen the waves on the surface. And they were noting the correspondence between the occurrence of the cold seeps and the occurrence of the corals, which is also being described in the Northeast Atlantic. Now in this case, we think that not very many of the corals are growing on the crusts, and we don't think that they're metabolically dependent upon the hydrocarbon seeps, but we will be trying to find that out using stable isotopes. And again, so I'm going to skip over that one. Back to the Lapthedia site. So Barbara showed you the picture of the coral bycatch, and I just wanted to show you something about these steep rock walls and the geomorphology of the site. So the year before we went there, a German ship, because this was such an exciting find, this Lapthedia site, a German ship had gone there and collected the multibeam data. And so this is the slope raster of that multibeam data in that area. The redder it is, the steeper the slope. And so this slopes up to 89 degrees. Pretty steep, okay? And so, the tracks here show you the distributions of the dives. So very, very steep rock walls, and they're all oriented in a certain direction. Barbara showed you pictures of what the corals themselves look like. Reef-like knolls, but growing on these vertical rock walls, facing into these strong currents. So my undergraduate honors student, Yuthika Jalim, did this for her honors, looking at the distributions of the corals by depth, but also with regards to orientation. And so the corals are all facing one specific direction to the south, into the southeast, where the current's coming from. And if you look at the other kinds of fauna in the other area, all the other types of sponges or other kinds of corals other than Lapthedia have a much wider range of distributions. So again, the interaction of the nature of the bedrock geomorphology in this case, and the biota living on them. Okay, and again, the important habitat for fish. Okay, let's change gears. A species that has been overlooked of corals in our waters is Acanella arbuscula, and this is a species that Laura Picarillo is working on for her master's degree. So thank you, Laura, for these pictures. This is a species that is very widespread, but is not very big and not very showy. Ironically, some of the fishermen say it stinks, and they've even reported feeling nauseous when it comes up in the nets. It's called a bamboo coral because it has these segments to its skeleton, so you can see the calcite internodes and the protein nodes. And if you cut open those nodes, you can see the growth lines. So this is under plain light. This is under fluorescent light, fluorescence microscope. So you can count the growth lines. We think the growth lines are annual, but we're trying to verify that. So with the help of a DFO grant, we were able to build these coral staining chambers and deploy them this year. So here's one of those small corals underneath. They're about to get stained. We go on. Gosh. Okay, so this is the calcine or calcine fluorescent dye, which is going to be in the chamber. The coral gets stained for about 6 hours, and then in 2 years' time, we'll go back and collect the very same coral. We've got markers out there, and we know precisely where it is, to verify the growth banding of those coral, of the growth bands. The reason we want to know that for sure is that it matters quite a lot how old these corals are in terms of their ability to recover from damage, including fisheries damage. So that's Laura's project. This is a muddy bottom, very different from most of what we've been seeing. Elsewhere, most of the coral habitats are typically pretty rocky bottoms. And so if you look at it, it doesn't look like very much. The corals are pretty small. They're mostly like 10 centimeters high and smaller. So not a very visually impressive environment compared to some of the others, but again, quite important. Oh. I'm going to skip this because we're late. Okay, Barbara mentioned the bamboo coral forest. So this is where we get back into the question of how long have these habitats been in existence? So one of the things we did this year was some piston coring and gravity coring in that site. That's the sound of the thrusters. We don't really like that. Okay. The important thing to note here in that video is you see all the mud accumulated between the corals. Think of a snow fence. So the snow fence is accumulating the snow, and then where you don't have a snow fence, you might have a barren area with almost no accumulation of snow there. The same thing with these corals underneath the water. They are accumulating the mud that is drifting past them in the current. So we took cores in 2016 and again this year to try to look at what's the age of these. So the large scale topography here is in the underpinning, but in the small scale topography is what we see with those corals accumulating the sediments. So you take a core, it's a piston or gravity core going down through the mud. The indications here are depth of the core in centimeters. You'll notice that all the little white things there are coral fragments. So you get down to about 130 centimeters depth in this core, and you see the transition from mud background to gravel, and there's a coral right at the base of that. The age of the coral, the radiocarbon age is 2,600 years, so it's approximately 2,000 calendar years old. And we can age the gravel elsewhere in a different core from the same location as being about 13,000 years. So two immediate questions there are, all right, is this site representative or not? Hence the need to collect more cores. But secondly, what happened there for 11,000 years? And why was there not any deposition of sediment? So that's a geological question we will not be able to answer here, because we don't have a record. And just to show you, again, this is what we would do with that, is developing an age model. I can't point with my left arm yet. Of the depth in the core versus the ages of the corals data. So a very consistent and continuous occupation of that habitat by those corals. Okay, last thing. There's a big difference between the eastern side and the western side of Baffin Bay. And in particular, that has to do with, it may have to do with water temperature, but also carbonate saturation. So here's an example of those carnivorous sponges that Barbara mentioned, which we think might be playing the same kind of role as corals typically. So working with Kamiko Azetsu-Scott at Bedford Institute of Oceanography, we've been looking at the carbonate saturation of the waters in the northern Labrador Sea and Baffin Bay. So these are the ages of the dates of earlier acquisitions. We sampled more water this summer. So it's a complicated diagram. This one shows you pressure equals last and telling you the depth on the bottom of each one of the sites. And then this is the diagram we're going to look at in more detail. This is the carbonate saturation value at the bottom in each location. So there it is, bigger. 0 or 1 means it's fully saturated. That is to say, anything under than 1, the water is undersaturated with respect to aragonite. So an aragonite skeleton is going to want to dissolve. Anything above that, if the coral can make a skeleton, it's not right away going to dissolve. However, it's still going to cost that organism energy in order to make those skeletons. There's a Lophidia site, 1.4, right in it's still in that tolerable zone. And again, looking at that transition across the Davis Strait sill, notice we're getting into less and less saturated waters as we go further north and west. So this may be one of the limiting factors for the distributions of corals in our area. Is that carbonate saturation. Okay, last thing. Scott, in the full of surprises, again, a glacial trough. And so one of the things I wanted to point out is explaining this trough is a very different kind of trough from Makovic. It's again, it's glacially carved, but what's happening at the margins of it? And so this is where the cold-seep is happening. Those cold-seeps are both causing authigenic carbonate formation, but they might also be causing local erosion. So this is zooming in on the very, very edge of that trough in about 200 meters of water depth, and a very steep precipice on the edge of that with these heavily eroded cemented sediments. It's not bedrock. It's this cemented, carbonate cemented sediment. Probably, well, we don't know its age. That's one of the questions. We'd like to go back and sample it to determine its age. The other interesting thing there is we can determine that there is local turbidite sedimentation happening from the top of that flowing down into the trough. So these troughs are fascinating areas with this rich biodiversity, but each one's a little bit different. So to understand it, we have to look at the geology and the oceanography and the biology. We come to the end of our presentation.